In this video, we are going to talk about organelles. So as I noted earlier, organelles are specialized structures within the cell that have very characteristic shapes and they perform very specific functions. So each type of organelle has its own set of enzymes that carries out specific functions, and it serves as a functional compartment for very specific biochemical processes. The numbers and types of organelles vary in different cells. This is dependent upon the cell's function. Organelles all cooperate to maintain homeostasis, even though they all have different specific functions. So first off, the centrosome is located near the nucleus, and it consists of a pair of centrioles and a pericentriolar material. So here in this right diagram, you can see the two centrioles and the pericentriolar material surrounding them. The centrioles are cylindrical structures, each composed of 9 clusters of 3 microtubules. You can see that in this diagram here. Surrounding the centrioles is this pericentriolar material, and this contains hundreds of ring-shaped complexes composed of the protein tubulin. These are the organizing centers for growth of the mitotic spindle, so these play a very critical role in cell division and for microtubule formation in non-dividing cells. So the main function of centrioles and for the centrosome is going to be division, so the process of mitosis. Microtubules are the dominant components of cilia and flagella, and these are motile projections on the cell surface. So cilia are numerous, very short, hair-like projections, and these extend from the surface of the cell. So you can see them here in the second diagram. They each contain a core of about 20 microtubules surrounded by plasma membrane. The basal body of the cilium is anchored just below the surface of the plasma membrane. Now, flagella is very similar in structure to cilia, but typically it's a lot longer, and usually it moves an entire cell. So as you can see here on a sperm cell, the flagellum is actually the part of the cell that is responsible for the mobility of the sperm cell. A cilium displays a very oar-like beating pattern. So you can see this here in the ciliary movement. It's relatively stiff during the power stroke, but a lot more flexible during the recovery stroke. So here going right is going to be the power stroke, left recovery stroke. Many cells of the respiratory tract have hundreds of cilia that can sweep foreign particles that are trapped in mucus away from the lungs. So this is very important in keeping your lungs free of mucus and free of a lot of other foreign particles and fluids. In cystic fibrosis, this is where the extremely thick mucus secretions are produced, and it really interferes with the ciliary action and the normal functions of the respiratory tract. So when we have the disorder of cystic fibrosis, this is when you have this super thick mucus. It kind of like covers all these cilia, so it keeps them from moving, and it keeps them from moving fluid and moving that mucus out of the lungs, and it keeps these particles kind of trapped in there. So it really interferes with the normal function of your lungs. Flagella, on the other hand, these generate forward motion by rapidly wiggling in kind of a wave-like pattern. As you can see here, this on the left side, this flagellar movement. So the only example of flagellum in the human body is a sperm cell's tail, which propels the sperm toward the oocyte in the uterine tube. So you can kind of see here, it's kind of a wave-like movement going up and down, moving the cell forward. Moving on to ribosomes. So these are the sites of protein synthesis. The name reflects their high content of one of the one type of ribonucleic acid, which is rRNA, or ribosomal RNA, you can see here. They're made up of two subunits, a large subunit and a small subunit that come together to make the functional ribosome. They're made separately in the nucleus, and then they exit the nucleus separately, and then they come together to create the functional ribosome in the cytoplasm. Ribosomes can either be attached to other organelles, such as the outer surface of the nuclear membrane, or to the extensively folded membrane called the endoplasmic reticulum, which we'll talk about later. Or ribosomes can also be free or unattached to any other surface in the cell. Free ribosomes will synthesize proteins that are used in the cytosol. So just write that down here. So free ribosomes means they're not attached to any other organelles. These make proteins that are used in the cytosol. There are also ribosomes that are located in the mitochondria, and this is where they synthesize mitochondrial proteins. So if they're in the mitochondria, sorry, rewrite this. If they're in the mitochondria equals mitochondrial proteins. The endoplasmic reticulum is a network of membranes that form flattened sacs or tubules, and it extends from the nuclear envelope, as you can see on this right diagram here. So here you have the nuclear envelope that's kind of in red, and the endoplasmic reticulum extends from there. It's in green. And it projects throughout the cytoplasm. It's very extensive, and it kind of makes up more than half of the membranous surface within the cytoplasm of most cells. There are two types of endoplasmic reticulum within cells. They are the rough endoplasmic reticulum, or just shortened to rough ER, and the smooth ER. So the rough ER is continuous with the nuclear membrane. As you can see here, you can see that it's very continuous with the membrane here. And it's usually folded into a series of very kind of tightly folded sacs. The outer surface is studded with ribosomes. You can see here these blue dots are going to be ribosomes on the rough ER, and these are sites for protein synthesis, like we covered in the last slide. Proteins synthesized by the ribosomes that are attached to the rough ER, they enter the space within the rough ER, and they are processed for sorting. So proteins that are made in those ribosomes are sent back into the ER, into the endoplasmic reticulum, and they are processed and sorted. So these are proteins like secretory proteins, membrane proteins, as well as many other organellar proteins. The smooth ER extends from the rough ER to form a network of membrane tubules. So these look a lot different than the rough ER, as you can tell. So the rough ER has a lot of like folds. So you can see here lots of folds, whereas the smooth ER is kind of looks like tubes or like a network of caves and caverns. So unlike the rough ER, the smooth ER does not have any ribosomes on the outside surface. However, the smooth ER does contain a lot of unique enzymes that make it extremely functionally diverse from the rough ER. So because there's no ribosomes on the smooth endoplasmic reticulum, it doesn't synthesize proteins, but the smooth ER does synthesize fatty acids, and steroids. And remember, these are steroids like estrogens and testosterone. Most of the proteins that are synthesized in the ribosomes that are attached to the rough endoplasmic reticulum are transported to other regions of the cell. So the first step in this transportation is the Golgi complex. And the Golgi complex is quite important. The purpose of the Golgi complex is to package and modify proteins in vesicles. So I'm going to say package and modify proteins. So the Golgi complex is made up of about 3 to 20 cisternae. So here you have a single cisterna. And cisternae are flattened small sacs with bulging edges that resemble kind of a stack of pita bread, I guess. The cisternae at opposite ends of the Golgi complex, they differ from each other in size, shape, and also their enzymatic activity. So here you can see this cis face on this side looks a lot different than these on the trans face. These are quite different looking edges. So this is the entry face, or the cis face. This is a convex surface, and this faces the rough ER. So you're going to have the rough ER kind of over here. And this is where proteins that are synthesized in the rough ER, this is where they enter the Golgi complex. So here you can see these transport vesicles that are coming from the rough ER, and they have proteins that have been synthesized on the ribosomes on the rough ER are inside those transport vesicles, and they come to the Golgi complex to be packaged and modified. There are different enzymes within each location of the Golgi, so it kind of depends on different areas. There's going to be different stuff located. It's not the same throughout. In the end, as it goes through each of these different cisternae, as we go through and further, and then we come out on the trans face. This is where these now packaged and modified proteins will exit the Golgi complex by secretory vesicles. And we're going to look at that in the next slide. So as we can kind of see a little bit better here, the cis face receives and modifies proteins that are produced by the rough ER. So over here we have the rough ER. It's going to be right, as you can see, it's right here. These are these synthesized proteins that are put into these transport vesicles that then reach the entry face of the Golgi complex. And they pass through the different faces, or the different cisterna of the Golgi complex. The medial cisterna, this is where carbohydrates are added to the proteins to form glycoproteins, and lipids to proteins to form lipoproteins. So that's where we're going to have added carbs onto these proteins. And we're also going to add lipids here too. The exit or transface, this modifies the molecules even further, and then it also sorts them and packages them to transport to their very specific destinations. So the transface actually faces the plasma membrane, and it's the final stop for proteins within the Golgi complex before they're sent to other organelles for use. So here we have a couple different examples of some vesicles. So here we have a secretory vesicle. So this is sent to the plasma membrane to be exported by exocytosis. We also have a membrane vesicle, and these are sent to merge with the plasma membrane and actually be stuck to the plasma membrane as a membrane protein. And then here we have another transport vesicle, and this is going to be going to a lysosome. So then lysosomes are organelles that are enclosed vesicles that form from the Golgi complex, and they contain many powerful digestive and hydrolytic enzymes. So that's going to be their main thing. They're going to have a lot of these digestive enzymes. Once they fuse to other vesicles that are formed during endocytosis, they work to break down those proteins that come in them in the form of vesicles. So we saw that when we talked about the 3 different types of endocytosis, and they fuse with the lysosomes, and then whatever's within those vesicles is digested by the digestive enzymes coming from the lysosomes. Since lysosomal enzymes really work best at a very acidic pH, so we want to have a very low pH. The lysosomal membrane includes these transport pumps that actually import hydrogen ions and allows the interior of the lysosome to actually have a pH of about 5. So we're going to import hydrogen ions. So we're going to have a pH of 5 approximately. So that's a lot more acidic than the pH of the cytosol, because the cytosol pH is going to be 7, so it's going to be neutral. So it's a lot more acidic than the cytosol. Pretty much the lysosomes are like the trash bins of the cell. So they take in damage or unneeded organelles, debris, or anything else that pretty much digests it completely, and then releases the usable molecules back into the cytosol. So it's going to release things like glucose, like amino acids, or like fatty acids as well. So these are going to be the product of the digestive enzymes. Okay, the mitochondria. This is known as the powerhouse of the cell. This is because most of the energy in the form of ATP is created within the mitochondria. And this is through aerobic respiration. And aerobic respiration means oxygen requiring. Aerobic means that O2 is needed. So there is an outer and an inner membrane of the mitochondria. There is small fluid-filled space between them. You have these extensive folds in the mitochondria. These are called cristae. As you can see here, mitochondrial cristae, these are what these folds are called, and they provide really large surface area for chemical reactions that are part of that aerobic phase of cellular respiration. So we're going to have cellular respiration occurring. And that's why we need that large surface area, because all those chemical reactions need a lot of space. So a cell might have as many as a few hundred mitochondria or even several thousand mitochondria. The mitochondria also has a very important and early role in apoptosis. Apoptosis, right here, this is going to be known as programmed cell death. And this occurs in response to destructive free radicals or DNA damage, or growth repair deprivation, lack of oxygen and nutrients, or also certain chemicals that are released from the mitochondria. So when this occurs, we have additional chemicals that are released from the mitochondria following the formation of a pore on the outer mitochondrial membrane. One of these is cytochrome C. So cytochrome C is actually the protein that initiates a cascade of activating these protein digestion enzymes that bring about apoptosis. Really, all you have to know is that cytochrome C is released by the mitochondria. That produces apoptosis, which means it's going to cause the cell to kill itself, essentially. So the cool thing about the mitochondria is that it also has its own set of DNA. And this is separate from the DNA that's found within the nucleus. So where the genes in the nucleus are inherited from both your mother and your father, the mitochondrial DNA is inherited only from your mother. So that's kind of interesting. The nucleus. This is a spherical or oval-shaped organelle that's usually the most predominant feature of a cell. Most cells have just a single nucleus, though there are a few exceptions. Red blood cells, they don't have any nucleus. So, red blood cells, no nucleus. Skeletal cells and some other cell types, or skeletal muscle cells rather, they have multiple nuclei, so that's pretty cool. There is a double membrane on a nucleus that encloses the nucleus, and it has pores throughout it. So these nuclear pores control the movement of substances between the nucleus and the cytoplasm. So here we have this nuclear pore, as you can see here, so it controls the movement of substances between the cytoplasm and the nucleus. You have small molecules and ions can move through the pore, and this is by passive diffusion. So this is a passive process, doesn't require ATP. But most large molecules like RNA and protein, they can't pass through unless they pass through by active transport. So passive diffusion, these are very, very small molecules or ions as well. Where if we have, by active transport, you're going to need active transport for RNAs and proteins because they're too large. And this is going to be transport through those pores. So within the nucleus resides kind of these hereditary units. These are called genes. And these can be condensed into chromosomes. The DNA is a double helix of linked nucleotides. So here we have this double helix, they're twisted around histones. So these are these histone proteins that will help to condense the DNA even further. So they're twisted around, as you can kind of see here. And then they're twisted even further into a chromatin fiber. They're kind of compacted further and further until they resemble this bow tie, which is called a chromosome. It kind of looks like a bow tie or a butterfly, something like that. So as you can see, they are extensively compacted. So within even this one chromosome, there is a ton of DNA. So among all your chromosomes, there is a lot of DNA held there. And that will be the end of part 6.